Hello everyone. In this video, we'll talk about the regulation of fatty acid synthesis in continuation of our fatty acid synthesis videos. Okay, so what are the requirements we have already studied? What are the requirements of fatty acid synthesis and what are the differences between beta oxidation and fatty acid synthesis also? So here the main coenzyme that is requiring for fatty acid synthesis, that is NADPH, and we have learned what are the sources of NADPH. The main source of NADPH for fatty acid synthesis is pentose phosphate pathway or hexose monoose phosphate pathway, HMP shunt, you can say, right? So tissues which are having like active lipogenesis, they should also have active HMP shunt pathway also to supply this NADPH. So HMP shunt mainly active in liver, adipose tissue, lactate mammary gland, as fatty acid synthesis actively taking place in these organs as well. So to talk about the regulation of fatty acid synthesis, so first any synthetic pathway, there should be availability of substrate. So what is the substrate? The substrate is acetyl-CoA here. Okay. So fatty acid synthesis and which conditions will be having acetyl-CoA? So more of glucose intake, okay, more of carbohydrate rich meal. So in that condition, the intermediates of TCA cycle are limited. So whatever the acetyl-CoA is coming from the glycolysis end pathway, okay, so it cannot be involved in TCA cycle. So the excess of acetyl-CoA which will be turned to alternative pathways such as fatty acid synthesis and ketone body synthesis. Okay, and there is again exception in which conditions ketone bodies will be synthesized. Otherwise, majority of acetyl-CoA apart from TCA cycle, they will be diverted for fatty acid synthesis. So fatty acid synthesis occurs when carbohydrate is abundant and the level of fatty acid is low. The availability of citrate in cytoplasm is the most important regulator factor producing short-term effect. So because citrate is the only way, because fatty acid synthesis is taking place in cytoplasm, okay, and the acetyl-CoA is in the mitochondria. So how to get out of this acetyl-CoA from mitochondria to cytoplasm is only way, that is citrate. So the availability of citrate also play a major role in fatty acid synthesis. And acetyl-CoA carboxylase, the enzyme, okay, the first enzyme, the rate limiting enzyme of fatty acid synthesis, that is acetyl-CoA carboxylase. So this rate limiting enzyme, how it regulates fatty acid synthesis? You see, phosphorylation inactivates acetyl-CoA carboxylase. That means dephosphorylated state and phosphorylated state. So two states are there. So the acetyl-CoA carboxylase is activated in dephosphorylated state and which promoted by insulin. But in case of glucagon, what happens? Phosphorylates acetyl-CoA carboxylase and inactivates the acetyl-CoA carboxylase. So AMP activates the phosphorylation while ATP inhibits it. That means what is indicated? AMP indicates there should be like energy. AMP activates the phosphorylation process. That means AMP indicates deficiency of energy. ATP indicates excess of energy. Okay. So they have the contradictory action over the acetyl-CoA carboxylase. So when we require fatty acid synthesis, so when we are of high energy, okay, high energy we are having, okay, we don't want any energy to produce, so no need to produce TCA cycle. So acetyl-CoA will be involved in fatty acid synthesis. AMP is indicating of deficiency of energy, so we require energy. So for that we don't want this fatty acid synthesis to take place. Okay, so fatty acid synthesis is enhanced when energy charge is high, okay, and fatty acid synthesis decreases when glucose level and energy charge is low. Okay, here so AMP indicates low level energy and low levels of glucose. Okay, ATP indicates high levels of energy and high glucose. So the enzyme is inhibited by palmitoyl-CoA, the end product. So it is called as feedback inhibition or end product inhibition. Because already we are having enough of palmitic acid, so again there is no need of further production of palmitic acid. So this end product will be inhibiting the acetyl-CoA carboxylase. Next, what are the hormones will be involved in this fatty acid synthesis? Acting as activators or like inhibitors. So the major hormones that we have to discuss here: insulin, glucagon. Okay, insulin, what it favors? It favors lipogenesis. So because it increases the utilization of glucose, so that way it reduces the blood glucose levels. Okay, when promoting the glucose oxidation, more acetyl-CoA will be forming. Okay, so as I told you, there is a limitation for the intermediates of TCA cycle. So not all the acetyl-CoA will be entering into TCA cycle. Some of the acetyl-CoA will be diverted for the fatty acid synthesis. So this way insulin is favoring lipogenesis or fatty acid synthesis. So insulin also depresses hormone sensitive lipase. So hormone sensitive lipase is the enzyme. What it will do? It breaks down the fatty acids. Okay. So that way whatever the fatty acid is producing, it will be broken down. So fatty acid insulin is also inactivates hormone sensitive lipase, which is there in the arterial walls of the arterial walls in the body. Cascade activation. So if you see the picture, you see here how the cascade is activating in like hormone sensitive lipase activation. So growth hormone, adrenaline, glucagon, noradrenaline, corticosteroids, adrenocorticotropic hormone, thyroid stimulating hormone, thyroxines. Okay, so all there in one side and insulin is in the other side. Okay, what it is doing? So all these things through cascade like cyclic AMP. Okay, cyclic AMP is acting like a second messenger. So this cyclic AMP, okay, is converting inactive protein kinase into active protein kinases. So this way, what will they do? This active protein kinases activates hormone sensitive lipase and increases the fatty acid breakdown. Especially you can say triacylglycerols or fatty acids which are being broken down, indicative for the increasing the beta oxidation. Okay, so now here insulin alone inhibit the enzyme, inhibit the formation of cyclic AMP. So this way there is no conversion of inactive protein kinase into active protein kinase, and if active protein kinase is not there, it cannot convert ATP into ADP. Otherwise it cannot activate inactive hormone sensitive lipase into active hormone sensitive lipase. So here what to say? Hormone sensitive lipase is inactivated in dephosphorylated state and active hormone sensitive lipase is activated in phosphorylated state. Here what insulin is doing? Insulin is dephosphorylating inactive hormone sensitive lipase and inactivating it. If there is it is removing phosphate group, so it will be inactivated. But other hormones like glucagon and other hormones, growth hormones and all, they increases the activeness of the hormone sensitive lipase by adding phosphate group via cyclic AMP formation. So as we say, glucagon inhibits lipogenesis. Okay, because when we need energy, that time glucagon will come into the action. So glucagon want free glucose. Okay, so it will not allow free glucose to oxidize. Okay, so there is no acetyl-CoA and there is no there is no lipid synthesis. Rather than whatever the glucose is oxidized will be straight away entering into the TCA cycle. So that's why the inhibitor of fatty acid synthesis is glucagon. Okay, they inhibit acetyl-CoA carboxylase. So we have already told. So acetyl-CoA carboxylase will be dephosphorylated state and phosphorylated state. Okay, so what they say? So keeping the enzyme in inactive phosphorylated state. Okay, so acetyl-CoA carboxylase is inactive in phosphorylated state. That means it adds, glucagon adds the phosphate group to this enzyme and keeps it inactive. Whereas insulin removes the phosphate group and makes it active in dephosphorylated state. But it is quite opposite in case of hormone sensitive lipase. These hormones are secreted when energy charge is low and AMP levels are high. Next, so regulation at gene level. Okay, a diet rich in carbohydrate which leads to stimulation of both the glycolytic and lipogenic pathways. So genes encoding glucokinase and liver, okay, and pyruvate kinase of glycolysis and ATP citrate lyase, cell carrier and fatty acid synthase, okay, so of lipogenesis are regulated by modulation of the transcription rate. So along with the glucokinase and liver pyruvate kinase, so we have already learned glucokinase will come into the action when there is a high concentration of glucose, and this exclusively present in the liver, both glucokinase and pyruvate kinase. Okay, so ATP citrate lyase, cell carrier protein, and then fatty acid synthase, all these things, enzymes involved also encoded by the genes related to the glucokinase and liver pyruvate kinases. So this way fatty acid synthesis regulated hormonal and enzyme level and at the gene level. Thanks for watching. Thank you.